Welcome, Christopher Mason.
Right. Well, good afternoon, everyone. It's a pleasure to be here. And I think a lot of you came to get your phone swabbed over the past few days. So I'm here to tell you what we've found and tell you about some of the predictions we've done based on what's on your phone. So knowing that I'm the only thing standing between you and afternoon cocktails, I will try to make this talk like an afternoon cocktail in one small swoop. But you can see here the enthusiasm that was just listed by Juan and Peter about this graph, which has been in many genetics conferences throughout the world, is an empirical measure of the excitement that I have as a geneticist. That means that every single day we've generated more sequence data than we have in any other day before. And so that actually means that today is the best day to be a geneticist because we have the greatest amount of data, the greatest predictive capacity, and the best ability to make actually predictions about what something is when you've sequenced it. So this represents also the fastest pace of technological change that humans have ever witnessed. So it's very exciting. You can literally jump out of bed in the morning as a geneticist because it is empirically the best day to be a geneticist. But that's for human genetics. There's, of course, more than one genome. In your body's cellular democracy, your human cells are a minority party. So if there was an election to see where you should move your body, you would always lose that election. You might end up in a bathtub full of yogurt, maybe rolling around on the ground outside by other bacteria, but you are a minority party of your human cells. They kind of look a little bit like this. You're covered in these bacteria in, on, and all around you. They represent a key part of your biology. And if they step onto a scale, it's about maybe 3 pounds of your body, 2 to 3 pounds of your body is bacteria. So if your diet is not really working, you can legitimately blame some of the bacteria. They have this weight on you. But they also contribute a lot of the biochemistry to your body. And so most of them are commensals, but of course there are some that can and do go wrong, which lead to antibiotic resistance and antimicrobial resistance, which is estimated to be one of the greatest threats facing humanity. By the year 2050, will likely kill more people than all of cancer combined. So this is a real key question as to where is antibiotic resistance? Where is it moving and how is it moving? And for the most part, we don't really know. We haven't been able to track antibiotic resistance as well globally until sequencing has come down to be cheap enough to do so. So you can see here what people do in the subways is they're generally terrified to touch any sort of steel railing because they're afraid of what might be there. In this case, I'm actually more afraid, I think, of what's on this weird paper towel of unknown provenance that's been used in a way that I did not approve of. You can see there. Also, someone was scared of a railing once, wore a lab glove on the subway. They brought it to the next level to bring up their defenses. But for the most part, we don't know, actually. And so I became a lot more interested in microbiology when I became a father. This is my daughter who clearly loves ice cream. And when I took her on the subway when she was about 3 years old, there was one time she was grabbing the pole. She straight up licked the subway pole. And I immediately became curious because there was clearly a microbiome exchange that had just happened. And I really wanted to know what it was. I'm a dorky academic. I go look things up and say, well, we must know what microbial species are in a subway used by 5.5 million people every day in New York City. But there had never been a single study of what's on the surfaces that are all around us. So what we needed was sort of a microbiome and a genome snapshot of an entire metropolis, or what the New Yorker called the Microbiome. So we set out to do this in 2013, published a first snapshot in 2015 of the entire New York City subway system to see what was present. And we found a lot of mysteries, including some new species, some things that had never been seen before. And this has since expanded to a global study to track a lot of genetic diversity. So we play the video, please. This is a large international project where actually one day a year, we wake up and just past the international date line, in a coordinated fashion, send out people to swab all of the planet. So if I could, I'd have one giant Q-tip to swab the whole planet, but I'm not that tall. So what we do is coordinate with many labs across the world to build this genetic snapshot of antimicrobial resistance and genetic diversity across mass transit systems, clinical labs, and what's present in public areas around the world. So this project uses a lot of sequencing and technology and mapping, and we thought, well, we're doing this all over the world. What if we brought this to Milken and see what's actually on people's phones? What are these mobile genetic elements on mobile devices that people carry with them? What do they have on them? So we want to build these genetic maps around the world and then also look at AMR markers, the antimicrobial resistance. And then also you can use this information to scan for new antibiotics actually from the sequence data by itself. So again, as many of you went to the booth, you could see we wanted to get a phone metagenomics profile of the Milken conference. Thank you all for all those who came to the booth to donate your phone, your mysterious DNA. This is what it looked like. We brought these samples back to the lab. Some of the people from the laboratory and here at the lunch staying up all night to extract, prep, and sequence the samples, get them through the lab. This ended up being about a 48-hour total protocol to extract, prep, sequence, analyze the data. got the sequence data back about last night, finished doing the analysis, and we ran 24 different computational and prediction algorithms on the sequence data to see what we could figure out about all of you out here in this audience. We get generating about 1 million sequence reads of DNA per phone. Okay, so what did we find? Drum roll. So it turns out that a lot of you are mostly bacterial, human, and unknown DNA is what sits on your phone. So you can see, actually, on average, a lot of you have about 15 to 20% of DNA on your phone that we have never seen before in all of humanity. You can see here. This sounds weird. This is actually fairly normal for a shotgun sequence profile. What's even more interesting, though, is there is one guy here at this conference who has 75% unknown DNA on his phone. And I would like that person to please come see me after the lunch, because we have some additional studies we'd like to do. We did get one person who had 67% animal and plant DNA on her phone, to which I think maybe she has some of this lunch probably on her phone right now, I guess, is what we could see. But these results have been posted online. I'll give you a link at the end of the talk. It's also on the cards you got when you signed up. So all the data, all the plots are all available online, and you can browse them all at your leisure. So, that was interesting. And I thought, well, what else might be driving some differences? So this is across all the first batch of 96 samples. We see that there's actually over 2,000 species that we detected at this conference, so we are not alone. There are a lot of other species here, mostly bacteria and some viruses and fungi, but mostly bacterial friends that are in us, on us, and around us. Again, not too surprising, but it's a good number there. And then we could also see, what do they look like? Does it look like skin bacteria, airway bacteria? Where's it coming from in the body? You can see here, this is a cosine similarity of what it matches for different parts of the body based on occupation. So it's interesting, most people's phones look like skin microbiome. That's expected. It's a skin on your phone. But lawyers, you can see, really we're an outlier here on the right. And we know that they have been kind of talked about as growing extra teeth already at this lunch conference. So we know they've always been an outlier. Now we know at the microbial level that's true as well. So I can go to the next point here. Oh, is this? So we also, there's a few people at the conference, about 5% of you actually are smokers, most of you are not. But here you can see that smoking actually has shifted the microbiome in a statistically significant direction and pushed all these species out to a different direction, which actually gives you more evidence that smoking is bad for you. So all of those of you who are smoking, you should probably stop, just a quick reminder. And then we have to look at other parts of the metadata. So what's driving diversity? Is it smoking? Is it children? We can see here the people that have at least 4 or 3 children had the greatest amount of microbial diversity on their phones. And so all those kids grabbing the phones, licking the phones, dropping the phones, there you go. So that's kind of expected. But we also look at as a function of age. If you're the older, wiser person, do you bring more diversity? The answer here is very much yes. And this is actually mirrors what's been seen in public studies. So the old people at the audience, bravo, you've got more diversity. Keep on trucking, let's say. The other thing we did see is, on average, several dozen antimicrobial resistant markers per phone, and a total of 560. This is the map of all of them across all phones, the first 96 phones. And so this sounds maybe alarming to have dozens of antibiotic-resistant markers on your phone, except that this is actually very common. Actually, even in old bacteria that are 4 million years old, they too can be resistant to antibiotics. And so one of the goals of this study and what you have all contributed to is an understanding of what baseline antibiotic resistance is. So this is part of what is essential about these data, is to put it into context. So for the last 2 sections, we looked also at plants and animals. We got other DNA on your phone. What else is there? We did see a little trace of some things like alligator occasionally on some samples. One person had a huge amount of strawberry DNA. Again, this sample, I encourage this person to please come see me. I want to talk to you about your eating habits. This person had a lot of pig DNA, either a pork or a bacon fan, I'm not sure. But you can see that right there. Also very interesting. Just got these data last night. And then finally, we also took the human DNA that's on your phone to see what else did it map to. So as all of you know, your DNA tells the history of your ancestors, and so does your phone. So most of the self-reported ancestry at this conference was Caucasian, so what we could do from the predictive algorithms is actually see what's your ancestry. And in orange, you can see most of the European ancestry was the most predominant actually ancestry that we could see from the DNA on your phones. And but you do get some admixture, though. All of us are, if you go back far enough, we're all Africans, and you can actually see some of the admixture of people coming from different populations around the Earth. And so that means, in total, we can see what you've been doing, what you've been eating, a little bit of your ancestry, and predict all that from your phone. So if you want to see more details, we're posting the rest of the data on this website, and all the 1,000 samples will eventually be posted in about 2 weeks. And I want to thank you all for donating your DNA to the study. Thank you for your time. Enjoy your lunch, and keep your phone clean. Thanks.
Okay, great. Thank you. All right, after that, I know that all of you are trying to decide whether or not you go to the next panel or go buy a new cell phone. You can do both. Have a great afternoon. We're actually going to give you about 20 minutes now between sessions to talk among yourselves. Come join us for those sessions. Come join us for one of the 3 closing sessions. We look forward to you at poolside at 5 o'clock for cocktails. And thank you, each and all of you, for making this such a successful conference.